Hello guys, today we are doing question 4, which is basic agricultural genetics. Let's roll on. 4.1, a homozygous brown colored ewe was mated with a homozygous white ram in a breeding program. The brown color A is dominant over the white color A. The ewe produced brown lambs, and in the second generation the ewes produced white lambs. Okay, so from this thing we are given the homozygous brown ewe. Okay? It has brown color and a white colored ram. Yes, white color. Okay, then we're gonna go to 4.1.1. It says indicate the phenotype of the parent. Then what is the phenotype? The phenotype is the what? Is the physical appearance of the organism. Okay, it is the physical appearance. The phenotype is the physical appearance of the animal. Okay, it's what we can see. Okay, it's what we can see and observe about the animal with our naked eye. Okay, and for that shade or for that color to be able to be visible, it is backed up by the genotype. The genotype is the genetic makeup. Okay, so the genotype is what's going to tell us what the phenotype is. For example, if the genotype is zygous dominant BB, then you can see, okay, then the color is black. If dominant B is for Chi, then you can see, okay, that means that homozygous dominant animal is the one that represents black color. Okay, right. So yeah, here they just want the phenotype of the parent. So as you can see here, they say the homozygous brown colored ewe was mated with the homozygous colored white ram in the breeding program. The phenotype is the color. How is the color? The color is brown for the ewe. So we write brown colored ewe. Okay, because that's what we can observe. That's what we can see with our naked eye. The physical appearance, right? And with a homozygous white colored ram. Okay, then that means what I want to say, brown colored ewe and white colored ram. Okay, brown colored ewe and white colored ram. 4.1.2 indicate the genotypes of the parents. As I have said, the genotype is the genetic makeup of that animal. It's the genetic composition of that animal. Is those letters, the capital letter and small letter. Okay, you can have a homozygous recessive or a homozygous dominant. A homozygous dominant is when we have two capital letters of a letter. Maybe let's say BB. Two capital letters of BB. Okay, that's homozygous dominant. Then homozygous recessive, that's when we have those small letters. Okay, that's homozygous recessive. Two identical small letters. Okay, to identify small letters. And for example, we can have small letter BB, two small letter BB, and that is homozygous recessive. And also we can have heterozygous. Okay, heterozygous alleles. That's when we have a capital letter B and a small letter B. Okay, making it a combination of both. Two alleles, the other one is dominant, the other one is recessive. The other one has capital letter, the other one has a small letter. That's what we're talking about when we're talking about heterozygous condition in the alleles. That's when we have small letter and a capital letter, okay, which is dominant. The capital letter, that's dominant. The small letter, recessive. Okay, then for this, let's see. An ewe, an ewe is a female, and the ram is the male. Okay, then you can see where they say homozygous brown ewe. Okay, homozygous brown ewe, sharp. Then we go to a second sentence. The second sentence is a brown color is capital letter A, which is dominant. Okay, that means what the A is a capital letter and it's a brown color. Then we go back to the first sentence. It says homozygous brown color. That means what we're going to have two capital letter A's, two dominant A's. Okay, of the allele. Sharp. Then we go to the recessive one. Okay. Then this second sentence, it says the brown color A is dominant over the white color. That means this brown color masks the condition of this white color. That white color cannot be visible because it is masked by the dominant allele A. The dominant allele says, okay, now I'm going to just be visible. Okay, I'm the only one who's going to be visible. The white color cannot be visible because it has this small letter A, and this small letter A is recessive. That means it is masked. So it is masked if there is a dominant allele. Okay, so that means the color of the what? Yeah, the color of the ram will be AA, represented by AA, which is homozygous recessive. Homozygous recessive. Alright. Then let's see. Now that we have our letters, then the genotypes is going to have the E with the homozygous dominant, which is capital letter A's, two capital letter A's, or we can say two dominant alleles. The one that those terms, two dominant alleles. Okay. But we're going to represent it by the letters, but the letter AA. Then we're going to say E, AA plus ram, A, small letter AA, then answer there. 4.1.3, the type of dominance that is demonstrated on this portion, and the type of dominance is complete dominance. Okay? And you say give a reason for the answer. You can say that when a dominant allele is crossed with a recessive allele for the same trait. A trait is a characteristic for the same characteristic, and the dominant allele must the recessive allele in the phenotype. Okay. That means now the recessive allele cannot be visible because the dominant allele says, no, I'm the one here, I'm the man here. Okay, I deserve to be visible, and one cannot be visible. Okay. So you can see complete dominance. For example, let's say there is a celebrity. Okay, there is a celebrity. Let's say you see a celebrity at the mall, a celeb at the mall. Then when you are seeing a celebrity at the mall, okay, you're going to be focused on that celebrity. Okay, and other people will not be visible. All the memories will be about that celebrity. What that means? That celebrity was dominant. Okay, that means that celebrity was dominant. Your friends or other people who don't have their memories because you're focusing on that celebrity, taking pictures with that celebrity. It means that celebrity was dominant. Okay, that celebrity was able to mask. Okay, was able to mask the physical appearance of all of those people that were at the mall. I don't know if my example is bad, but yeah, that's how it is. I'm trying to be in real life situation like here. 4.1.5. Imitate the possible genotypes of the first crossing with the Punnett square. So for this crossing that needs Punnett square, it's not that complex, such as the crossing that is done in life sciences. Because with life sciences, they want you to just have all of those step-by-step crossing, like you have to show your parents. Your parents is black crossed with the white. Then you have to show the genotype of the parent D and the genotype of the other parent D. Then you have to show meiosis, meiosis, you have to show the gametes separating, to show fertilization, you have to show the actual crossing when it falls and the offspring. So, but here, when I see here, it is 4 marks, and the 4 marks is for the Punnett square. They focus on the Punnett square. That means if you can master the Punnett square, but also need to have the background knowledge of what is happening for you to arrive at the Punnett square. You need to cross the parent. You need to know the phenotype of the parent. The phenotype of the parent. Here we have the brown colored and the white colored. Yeah? And the genotype of the parent, we need to know the genotype of the parent. Okay. So a limb is an offspring, okay, or we can say it's a progeny. Progeny is the same term as the offspring, but sometimes they use progeny. Progeny just means offspring. Right. Then the E, as we have said previously, the E is represented by the two capital letters AA which is dominant, which is homozygous dominant allele. Okay, this is because brown is dominant. Okay, it is homozygous dominant, hence it has 2 capital letters. Okay, hence it has 2 capital letters. Then RAM, it has this phenotype, small letter AA, okay, which is homozygous recessive allele. This is because white is homozygous recessive, as I've said, and we use 2 small letters to represent this. Yeah, we use 2 small letters to represent this. We can see that the bull is homozygous dominant, right? So far we can see where the bull is homozygous dominant. This bull is a pure breed, okay? Pure breed. Okay, then the Punnett score is going to be like this. Where you're going to have your gametes, this side on the horizontal side and on the vertical side. The male gamete is going to be on the horizontal side at the top, and the female gamete is going to be on the left hand, on the left hand side, on the left hand side, vertical to A and B. Then from there you have something like this, and also we have 2 names here. I said name, and I show this symbol name, gender, and here I said female, and I show this symbol of the female, gender. Then A, dominant allele A and dominant allele dominant allele A and a recessive allele A, they give us a heterozygous condition, which is capital letter A and a small letter A. Then another one is capital letter A and a small letter A, is going to give us a heterozygous condition also, which is capital letter A and small letter A. And go to the third box, then capital letter A, okay, and the small letter A gives us a heterozygous condition, which is capital letter A, which is an offspring with capital letter A and a small letter A. Then we take the 4th box, we take 1 dominant allele from the we take 1 dominant allele gamete from the parent male, the male parent. Then we take another recessive allele from the female. We take this gamete from the female. Then when we mix these 2 alleles, they give us a heterozygous condition, okay, which is capital letter A and a small letter A, right? Is a heterozygous offspring. As you can see, for box 1, 2, 3, 4, all of our offsprings here have a heterozygous condition. Okay, have a dominant allele and a small, have a dominant allele and a recessive allele, which is a heterozygous condition. Then, the phenotype is that they are all brown. That is the phenotype. Okay, they are all brown, As you can see, I have 4 capital letter A and small letter A. Then yeah, 4.2.1, it talks about the crossing to improve the head. Yeah, 4.2, let's see. We have a scenario here. The table below gives information on an unknown breed of cows that were mated with pure breed Osmara bulls. The female offspring of the cows were always bred with the pure breed Osmara bull, as shown in the results below. Okay. Then you can see that the bull is homozygous dominant. Okay, is homozygous dominant. This bull is a pure breed with good desired trait, and it is mated with an inferior breed of cows. The aim is to improve the head of poor traits. Okay. The aim is to improve the head of poor traits until good quality offspring are produced, or until we provide the offspring with desired trait. Okay, let me repeat that. The aim is to improve the head of poor traits until good quality offspring are produced, or until we produce offspring that have good desired traits. So you can see that from course 2 until course 8, the head has improved. Has improved from 50% to 99.22%, which is nice. So this tells us already all of these things. They're trying to show us this is how they improve the head. Okay, this is how they improve the head by taking a pure breed and mating it with an inferior cow to improve the head. So let's see. 4.2.1, identify this type of breeding system. This type of breeding system where we made a superior bull with good traits with inferior cows. Then we crossbreed it from class 1, 2, 3, 4, 5, 6, 8 until we produce the offspring with good quality trait. What is this breeding system? This breeding system is upgrading. It's upgrading. 4.2.2 is the 2 disadvantages of this type of breeding system. The disadvantage of this breeding system is that we have to breed many times, okay, which is time consuming. For example, here we had crossing 1, 2, 3, 4, 5, 6, 7, 8 until we get the desired trait. The trait is a characteristic, until we get the desired characteristic. So this process is time consuming because we have too many breedings. We have to breed as many times. We have to breed as we have to breed again and again, and this is time consuming. Okay. Then another disadvantage is that it is expensive, as those have to be bought from outside to reduce inbreeding, because we have this herd of cows, and this herd of cows have inferior characteristics. Okay, so you have to get a bull that is outside of the herd. Okay, and that means we have to buy that bull or rent that bull or rent it. Okay, because if you take the bull that is in your head, that means you'll be doing inbreeding. So we want to avoid inbreeding or reduce inbreeding. That means you will have to rent or buy external bulls to come and improve the herd with those superior desired characteristics. Okay? We want that bull to bring those superior quality characteristics in our head. Okay, yeah. So that's why I say it is expensive as bulls have to be bought from outside to reduce inbreeding. We don't want inbreeding, that's why we take outside bulls. Brahmani. Oh, this is Bosmara. Yeah, this is Bosmara. 4.2.3 determine the number of courses needed before the first purebred Bosmara cows can be registered. Okay, we will have to have 5 courses. Yeah, you have to have 5 number of courses. Yeah, here at 96.87%. Yeah, 5 crosses. 4.2.4 calculate the percentage of purebred characteristics in the offspring in the third crossing. So for this question, step 1, okay, step 1, we're going to take the percentage of the cow, okay, but on the third crossing, that's where we have 75% and 100%. So we're going to take the percentage of the cow, okay, which is 75%, then we're going to plus it with the percentage of the bull, which is 100%. Then we're gonna divide it by 2, because we have 2 animals. Then we divide it by 2. Step number 2, then we'll get the answer from that 75 plus 100 divided by 2 is going to give us what? 0.875. Okay, then we take that 0.875, then multiply it by 100 to get a percentage answer. Then we end up having 27.5%. Then for that we get 4. Open drive, the table below indicates values. Heritability is 60% for birth weight, 33% for post-weaning weight gain, and 50% for post-weight. Then below they say a lamb's birth weight is 3 kg and the average birth weight for the flock is 1.8 kg. 4.3.1 determine the estimated breeding value EBV. Calculate the EBV for the birth weight. So calculating the EBV for the birth weight, you have to get your lamb weight. What is our lamb weight? Our lamb birth weight is 3 kg. We take this 3 kg. Then we take the lamb birth weight minus it by the average flock weight. Here our average flock weight is 1.8 kg. Okay, we take the lamb weight minus by the average flock weight, which will be 3 minus 1.8. Okay, then it's going to be 1.2 kg. Okay, 1.2 kg is what we got from subtracting the average flock weight from the lamb weight, 1.2. Then we take this 1.2 and we times it by the heritability. You can see it by the heritability. Okay, you can see it from the table. Okay, the table says birth weight, and under birth weight we have heritability, okay, which is 60%. We're going to take this 60%. Then we take, which is step number 2. Now we take 1.2 times the heritability of the birth weight, which is 60%. Okay, then we're going to get the answer, which is 0.72. Up to so far, you can see all we're doing there. Then 4.3. What is the heritability of the fleece weight of a lamb? The heritability of the fleece weight of a lamb. Okay, the heritability. Oh, I think I've skipped something. 4.3.2. Yeah, 4.3.2. What is the implication of the value in 4.3.1? What is the implication of this 0.72, the EBV for the birth weight? So the implication for this is that this means that the lamb's birth will weigh at 1.2 kg heavier than the average for the flock. This means that the lamb's birth will weigh at 1.2 kg heavier than the average for the flock. Yeah, then 4.3.2. What is the heritability of this weight of a lamb? The heritability of this weight of a lamb. So this question doesn't want you to calculate. Okay, as you can see, they say what is the heritability of the fleece weight of a lamb. Okay, so they don't want you to calculate, they just want you to find the value of the fleece weight, of the heritability of the fleece weight. Okay, heritability of the fleece weight. So we're going to go to the fleece weight on our table. Here's the fleece weight. Then the fleece weight has the heritability of 50%. This 50%. Then we write this 50%. That's what they want. 4.3.4 give 2 reasons why the post-weaning weight gain in the table cannot be recommended for breeding purposes. Okay, why the post-weaning weight gain in this table above cannot be recommended for breeding purposes. Okay, so we should indicate that the post-weaning weight gain has a low heritability. We have to indicate that the post-weaning weight gain has low heritability. There's a low heritability of 3%. This makes the environment have a huge influence on the outcome of the characteristics. Okay. Because of that low heritability of 33%, this makes the environment have a huge influence on the outcome of the characteristics. Okay. Then, yeah, let's roll on. Open for GM lettuce, GM genetic modification, right? Yeah. GM lettuce with a high yield was produced using a gene from a water plant. An experiment was conducted to test the effect of this genetic modification on lettuce plants. Scientists grew one group of plants consisting of GM lettuce and non-GM lettuce in a greenhouse, and a second group in an open field. The results of the experiment are given in the bar graph below. As you can see, the bar graph, this greenhouse and open field is the growth condition. Then we have the black for GM, the gray for non-GM. On our y-axis we have the yield to kg. The holding is the yield of GM and non-GM which is growing under different conditions. Okay. Right. 4.1.1, 4.4.1. Explain the difference in yield of GM lettuce and non-GM lettuce growing in the greenhouse and open field respectively. Right. The GM lettuce has more yield in kg. You know, it has an extra 10 kg in yield than the non-GM. Okay. When grown in a greenhouse and open field, the GM lettuce yields more better than non-GM, which is what is grown in different conditions. Okay. The GM lettuce has more yield. Okay. This GM lettuce has more yield in kg than the non-GM lettuce. Okay. And when grown in greenhouse and open field, the GM lettuce yields more better than the non-GM lettuce when grown in different conditions. Right. 4.4.2 deduce from the graph one advantage of GM lettuce for the farmer under both growing conditions. That means that they want the answer to be related to the graph. Deduce. Take from the graph. Deduce. Take from the graph. So it helps to increase the yield by an additional extra 10 kg. Okay? It helps to increase the yield by an additional 10 kg, which makes it to yield better. 4.4.3 identify 3 benefits of genetic engineering over traditional methods as depicted in the scenario above. The 1.3, okay. Plants are more resistant to diseases and droughts. That's point number 1. It increases the yield. So we can see an increase in yields in crops. Okay, can can see the increase in the yields for the crops. Point number 3, desired genes are transferred quickly or faster. They're transferred quickly because compared to other methods in traditional ways for improving the crops. It takes time to improve the crops using traditional methods, but with this genetic modification thing, okay, those desired genes can be transferred quickly using the various techniques, okay, of genetic modification. Then it is faster and quicker. And 4.4 suggests 2 potential environmental risks posed by genetically modified plants. 2 potential environmental risks posed by genetically modified plants. The creation of weeds. The creation of weeds can kill beneficial insects, disrupting food chain. So this GM plants and GM crops can kill beneficial insects. Okay, because the insect resistant crops, so they can kill beneficial or useful insects instead of killing those bad ones. So they can mistakenly kill the beneficial ones, and also they can create superweed. Creation of superweed. That's it.